Hi everyone, this is Lingana Gugulot, your pharma student, and today we are going to discuss about the receptor pharmacology in pharmacodynamics. So don't be late, let's get into the video.
Receptor pharmacology. The receptor pharmacology is a branch of pharmacology that focuses on the study of how drugs interact with the specific receptor in the body. In simple terms, the receptors are protein molecules located on the surface of the cell or within the cell, and they play a crucial role in transmitting the signals and mediating the effects of various molecular, including drugs.
In previous video, I have explained about the what is pharmacodynamics, principles of drug action, and mechanism of drug action. If anyone has not seen that video, for your reference, I am providing the video link in the description.
Here we go for the mechanism actions of receptors. The mechanism action of receptors which deals with the how drugs binds to the receptor and induce a biological response in crucial in pharmacology. And also it involves the studying of signal transaction pathway, second messenger system and downstream with the effects of triggered by the receptor activation. So, the classification of receptors. The receptors are classified into several types depending upon their structure, function, and mechanism. There are mainly four types of receptor. First one is G protein coupled receptor, second one ligand gated ion channel, third one enzyme linked receptor, and fourth one nuclear receptor. So, we go for the first one, G protein coupled receptor. GPCR is a family of large cell membrane receptor and which is activated by G protein, and this receptor is made up of 7 helical spanning membrane and which are interconnected to each other. The molecule of 7 alpha helical membrane spanning hydrophobic amino acid which runs into 3 extracellular and 3 intracellular loops. The G protein also float in the membrane where their exposed lying in the cytosol, and its heteromeric in composition consists of alpha, beta, gamma subunits. And the activation of these receptors leads to the displacement of GDP by GTP, and the activated alpha subunits carrying GTP dissociates from other two subunits and inhibit the effectors. The other rest two beta and gamma dimers also activates the receptor and operates the potassium channel to inhibit the voltage gated calcium plus 2 channels and promote the GPCR desensitization at higher rates of activation. A number of G protein is distinguished by their alpha subunits, and they are so important ones with their actions on different effectors. First one GS adenine cyclase activation, calcium channel opening, and GI adenine cyclase inhibition, potassium channel opening, GO calcium channel opening, and GQ phospholipase C activation. Here, a limited number of G protein are shared between the different receptors, and one receptor can utilize more than one G protein. Examples are GS is the coupler for the histamine H2, serotonin 5-HT4 to 7, glucagon, thyrotropin, and many more hormones. And GI is utilized by the opioids, cannabinoids, and some other receptor. And further, the alpha subunit has GTPase activity, and the bond of GTP is slowly hydrolyzed to GDP. Then the alpha subunit dissociates from the effector to rejoin its another subunit, but not before the signal is greatly amplified. Then the rate of GTP hydrolyzes by the alpha subunits, and the period for which it remains activated is regulated by the another protein is called regulator of G protein signaling. Moving to the three major effector pathways through which GPCR functions. First one, adenylyl cyclase CMP pathway, phosphorylase C, IP3 drag pathway, and C channel regulation. In adenylyl cyclase pathway, activation of AC results in the intracellular accumulation of molecule of second messengers, and CMP in which functions mainly through the CMP dependent. The protein kinase phosphorylates and alters the function of many enzymes, ion channel, transporter, transcription factors, and structural protein. And it manifests the contractility of heart, relaxation of smooth muscles, glycogenolysis, lipolysis, and hormone synthesis, etc. In addition of CMP directly opens into a specific type of membrane called calcium channel, called cyclic nucleotide gated channel in the heart, brain, and kidney.
And the second one, phospholipase C. The PLC catalyzes the hydrolysis of phosphatidylinositol 4,5 biphosphate in two second messengers. First one, inositol 1,45 trisphosphate, and second one, diacylglycerol pathway. These two play a crucial role in intracellular signaling. First one,
inositol 1,45 trisphosphate.
act on the IP3 receptor which are located on the endoplasmic reticulum and causing the release of calcium ions into the cytoplasm, and this elevated calcium ion levels which triggers the various downstreams signaling events such as muscle contraction, enzyme activation, and neurotransmitter release. And second one, DAC pathway. This DAC pathway activates the protein kinase C, which phosphorylates the target proteins involved in diverse cellular functions, including metabolism, growth, differentiation, and apoptosis. Third one, channel regulation. The activated G proteins like GS, GI, GO can also open or inhibit ionic channel specific for calcium ions and potassium ions without the intervention of any secondary messengers like CAMP or IP3, and brings about the hyperpolarization, depolarization, changes in the intracellular of calcium ions. For example, the GS opens calcium channels in myocardium and skeletal muscles, while GI and GO opens potassium channel in heart and smooth muscle, as well as it also inhibits the neuronal calcium channels. Second one, ligand-gated ion channel. It is also known as ion channel receptors, and it encloses the ion channel for sodium channels, calcium channels, potassium channels, and chlorine channels within their molecule. And these receptors are usually in pentameric structure of all the subunits in addition to large and extracellular segments. Generally, they have four membranes spanning helical domain, and these subunits are arranged in the channel in a round like rosettes. And these the alpha subunits usually bearing the binding site. In this, the agonist binding opens the channel and causes the depolarization, hyperpolarization, changes in the systolic ion composition. Depending upon the ion flows through the nicotinic, cholinergic, GABA A, glycine, glutamate, and 5H3 receptors. And thus, in this receptor, thus the agonist is directly operates the ion channel without any intervention required of coupling proteins or secondary messengers. And third one is enzyme linked receptors, and it is commonly known as catalytic receptor. These are a class of cell surface receptors and that possesses the intrinsic enzymatic activity are associated with the enzymatic domains. This receptor plays crucial role in transmitting the extracellular signals across the cell membrane and also initiating the intracellular signaling cascades. And these are a class of receptors and which are primarily utilized by the peptide hormone and made up of a large extracellular ligand. Typically, the hormone growth factor can attach and thus stimulate the enzymatic activity inside the cell. Here, most enzyme linked receptors are of tyrosine kinase type, which simply means that they display kinase activity and that a amino acid tyrosine involved in. So, when a ligand binds to this receptor, it causes the conformational changes and that results in the aggregation of both receptors. Once the dimer is formed, that tyrosine region and it's get activated causes the ATP becomes to ADP, which results in the phosphorylation of the receptors. Now, once of each tyrosine picks up the phosphates, then the proteins comes up and attach themselves to phosphorylated tyrosine. This results in the conformational changes in the attached protein, ultimately leading to the cascade of activation and that produces the cellular response. Fourth one is nuclear receptor. Nuclear receptor are a class of intracellular receptor found within the cell nucleus, and they also act as transcription factors by regulating the gene expression in response to the specific ligand. And these are a class of nicotinic receptors which are responsible for the sensing of steroids, thyroid hormones, and vitamins. These receptors are works with together with other proteins to regulate the expression of gene, thereby controlling the development of homeostasis and metabolism of the organism. These nuclear receptors also have a ligand binding domain that's bind to a specific lipophilic ligand such as hormone, vitamins, and metabolite intermediates. And this nuclear receptor also possesses a DNA binding domain and that allows to bind a specific DNA sequence called hormone response element. Upon the ligand binding, the nuclear receptor undergoes the conformational changes and that facilitate the recruitment of co-regulatory proteins and initiate or repress the transcription of target genes, thereby modulating the cellular process. And this transduces the signal by modifying the gene transcription. Example, glucocorticoids, mineralocorticoids, and vitamin D and E. I hope everyone liked this video and thanks for watching. Please like, share, subscribe to Pharma Student.